Cruising is unlike anything else. Our 3 cruise lines travel to every corner of the globe, providing exceptional guest experiences to see, understand, and appreciate the world from a whole new perspective. It's at the heart of what we do. The future of our business is dependent on the beauty and wonder of our planet.
Our Sail and Sustain program is our commitment to cruising responsibly and protecting the oceans we love. We focus on making a real difference both at sea and on shore, because cruising depends on healthy oceans and thriving communities. It's how NCLH shows guests, employees, and investors that we are serious about reducing our footprint, creating value for our investors, meeting global sustainability goals, and staying ahead of regulations.
Welcome. Thank you all for coming. For those of you that haven't come to Sea Secrets this year, I'm the new dean at the Rosenstiel School. My name is Ben Kurtman. And so if anything goes wrong tonight, you can blame me. And everything that goes well, Deanna Udell gets all the credit. This is our last program of 2026. So, but don't worry, there'll be something next year too. Don't be concerned. And I want to thank you all for joining us, all of you that are here in person, and all of those that are online. Welcome. I do want to take a moment to thank all of our sponsors. Of course, you saw that lovely video from our presenting sponsor, Norwegian Cruise Lines. That's the 2nd year they've been our presenting sponsor. Our supporting sponsors include the Shepherd Broad Foundation, Bill Galway, who's here, and we'd be more than happy to chat with you about anything related to anything. The KB Life Enhancement Forum, the Key Biscayne Community Foundation, the Joan McCon Family Foundation, Nicole and Myron Wang, and Southern Glaciers Wine and Spirits. We thank them all for supporting our program. This is why we can continue to do this fantastic Sea Secrets program. In fact, yes, thank you, Dennis. When I was considering taking this job, I told some of the university leadership that I'm embarrassed to ask my wife for $10 for lunch. So I'm not very good at asking for money. I said, don't worry, Ben, you never have to ask for money. However, I have to ask for money. So a priority of mine, of course, if you've enjoyed joining us for Sea Secrets and learning about our programs and hearing from our presenters, I welcome you to join us in being a supporter if you have not already done so, and a hearty thank you for those that continue to support the Sea Secrets program. That's it. That's all I got for asking. That's the best I can do. And if you want to give me $10 for lunch, I'd appreciate it. A priority of mine is to really grow the way I think of it is our hands dirty, feet wet kind of learning that we do here, experiential learning. And you're going to hear a little bit more about how important that is from an undergraduate student that's just about to graduate. So Paige Fitzpatrick is going to talk in just a minute, Paige. She's going to talk about a few things, and I think this is going to give you a sense of why I think this hands dirty, feet wet kind of learning is so important for attracting the best and brightest here. And so that's really where we're pushing a lot of our efforts. And if you want to hear more about that, more opportunities to participate in that, Jennifer, where are you? There she is. Jennifer, please stand up. Jennifer is the one that you should talk to. So Paige, you're on.
Thank you. All right. Hi, everybody. Thank you so much for having me here today. My name is Paige Fitzpatrick. I am an undergraduate student here at the Rosenstiel School, studying marine affairs. And today I'm going to be talking to you guys about my hands-on experience training in marine technology at sea. So the reason I first chose the University of Miami was because I had this dream to become a seagoing marine scientist. And the university has so many incredible opportunities to train both in the classroom and outside of it. So many different laboratories and opportunities for me to really get my hands dirty. So as my time here went on, I realized that my niche within the marine science industry is as a marine technician, focusing on marine technology at sea. So you guys might be wondering, what is a marine technician? Which is totally fair. In the marine research world, a marine technician on board a research vessel is the interface between the science operations and the shipboard operations. So it's very interdisciplinary, and you need a lot of different skills to be able to fill this position. A lot of them can be learned in the classroom, but a lot of them need to be trained, and working with mentors is a super good way to learn these skills. So after I realized that I wanted to be involved in marine technology, I reached out to pretty much all of my professors at the university and got directed to Dr. Dennis Hansel, who directed me to Cedric Guigand in the Makers Lab. The Makers Lab is our marine technology fabrication laboratory, which does a lot of really cool work with 3D printers, which you can see in the photo there. I was also directed to the Sustain Air-Sea Interaction Laboratory, which is our hurricane wind and wave tank. So in these labs, I received direct mentorship from the chief scientists and engineers, and really got a lot of experience with hands-on learning and developing my skills. I was able to apply these skills to an internship on board the RV FG Walton Smith, which is our research vessel with the school. I worked as an intern for a summer, working in the marine technology department, and I learned a lot of really great skills and met a lot of really great people in this position. I ended up getting hired on board the vessel after my internship as an assistant engineer and an assistant marine technician. In this position, I was able to work on a lot of really cool equipment, including what's called the CTD. That is a deep water sampling device, which is pretty essential to any oceanographic research vessel, and I was very glad to get some hands-on experience learning with that equipment. I was also able to work within the UNOL's research fleet. So that's the fleet of academic research vessels in America. And through this experience, I was able to find a really incredible community of marine technicians that helped support me on my journey, both at the university and afterwards. So through this experience, and you'll have to apologize, I look very sunburnt in that picture, but I was able to conduct my own research at sea through a grant fund through the university. I was able to test 3D printed instruments under hydrostatic pressure, so basically sending them down to the bottom of the ocean and seeing if they could withstand the pressure. I was able to use my skills in this marine fabrication realm, as well as my research, to present at a national conference, which is what you see over on the right-hand side. That is the RV Tech 2025 conference out at Scripps. So with my sea days on board the Walton Smith, I was also able to go for my 100-ton U.S. Coast Guard captain's license, as well as my QMed marine engineering license. This is me running small boat operations on board, and we were doing a dive operation in the Bahamas, so really great experience. So what's next for me in this industry? After my graduation, I'll be joining the RV Sekuliak with the University of Alaska Fairbanks to train on their multi-beam sonar system. I can confidently say that this university has done more than enough to prepare me to take the next steps in this industry and to make that dream that I had of becoming a seagoing marine scientist a reality. Thank you.
Thank you, Paige. There's hope for us yet. Thank you very much. Okay. So next, I'm supposed to introduce Gabriel, today's speaker. And as usual, they've given me a paragraph describing Gabriel's background, and I'm going to flub it up because it just doesn't do Gabriel justice. So here, let me read, and then I'm going to add a little color to it. Gabriel Reygondeau is a marine biogeographer and an associate professor at the University of Miami's Rosenstiel School's Department of Marine Biology and Ecology, and a core faculty member of the Frost Institute for Data Science and Computing. What that means is Gabriel sort of spends half his time at thinking about computational issues and half his time thinking about marine biology and ecology, and he's trying to marry those two problems. And so here, I'm getting off script. And so normally at this point in time, I would say that Gabriel is mapping all the fish in the world's ocean, and he would groan. See, he just groaned. And why does he groan? Because that's not ambitious enough. That's not ambitious enough. He's mapping all of the marine life in the world's oceans. It's amazing. So when people ask me, what does this guy Gabriel do? I say he's mapping all of the marine life in the world's oceans. That is phenomenal. Absolutely phenomenal. So Gabriel's going to tell us about that. He grew up in he's grown up in 25 different countries around the world. And that has really fostered his early fascination with nature, the ocean, and the diversity of life on Earth. With respect to diversity, I think I got the year right on this, Gabriel. 2019, you were labeled the marine guy in the marine guy for obvious reasons in the Half Earth Project. So what's the Half Earth Project? The argument here with the Half Earth Project is that in order to preserve 85% of the world's biodiversity, we need to protect 50% of, we have to have 50% of the world in protected habitats. So the challenge is, what are the 50%? Where's the right places? And so Gabriel was the guy that was going to solve the problem for the entire marine life. So small, just a unambitious guy, right? So anyways, this was part of a really interesting postdoc too, as far as I understand. It was a collaboration between UBC, University of British Columbia, and Yale. So he was going back and forth between those places. Then he spent a significant amount of time at the University of British Columbia, UBC, where he continued to develop biodiversity tools, and of course, Gabriel's extremely modest, and so the other thing he won't tell you about is, and I'm going to choke this, I guarantee it. UTMB, Ultra Trail du Mont Blanc, right? So this is of course very unambitious. It's like a 175 kilometer jog on a trail in a mountainous region, right? And so they rank you guys, right? You get, yeah, it's like the most elite of the elite. Clearly. Somebody needs to sort of coach him on some ambition, for goodness sakes. Anyways, Gabriel is going to tell us all about his efforts to map all of marine life in the world's oceans. I can't tell you how excited I am about this, Gabriel. Thank you for doing this.
Hello, everyone. I would like to start with this picture. This picture was, and I think it's really apropos here, especially with Artemis just coming back from the moon. This picture was taken in 1990. In 1990, we had a satellite going across our galaxy named Voyager 1. And Carl Sagan, I'm pretty sure half of you have heard about Carl Sagan, asked, can we turn the camera and just look at where we are? And the pale blue dot that you're seeing is us. You need to imagine this pale blue dot is everything you know, all life form that we know. We are a statistical anomaly of the universe. In theory, there should be life everywhere. But from what we know, this pale blue dot is the only place where there is life. Sorry. And when I'm showing you this world rotating, we are inherently, you see, I told you the age is terrible, terrestrial creature. When you look at that map, in 5 seconds you will see a desert, you will see a forest, you will see delineate different ecosystems, because we have been trained, because by evolution we have been trained to recognize different types of ecosystem in the world. And therefore, in history, we have tried to understand what was surrounding us in this pale blue dot. And I take this journey because this is the most famous one. I'm pretty sure you have seen it. This is Darwin on the beagles, trying to go around the world and trying to describe what he saw and write a book not really known, not just the biggest citation in the world after Einstein, The Origin of Species. Since then, taxonomists and biologists and ecologists have continued to describe. So what you're seeing here on the right is on the x-axis the year of a discovery of a new species, and on the y-axis the amount of species. And this is only for terrestrial vertebrates. And what you can see is that before the 1950s, so pre-Second World War, most of the vertebrates were already described. And now every discovery on land is kind of a tiny bit more. And so already pre-Second World War, we knew where was the hotspot on the terrestrial. We knew what was this key space on land that we need to protect, that we know that there is something specific from the Horn of Africa to the Mesoamerica to here with Florida and the Caribbean islands. And I'm not going to do all of them. What you need to remember is that we have more species in the tropics than we have in the pole. But doesn't mean that the poles is less important than the tropics because of rarity. And I really love that quote. I started my PhD thesis with this quote. What about the ocean? For the uneducated eye of a non-sailor, 3/4 of the planet is basically pale blue. And all the technology, all the new sensors, all the AI is just giving us a new set of glass that allows us to see the invisible frontier that we couldn't see before, to see the ecosystem that we didn't had an idea before. To give you an idea, the French philosopher of the 1800s thought that the marine ecosystem was the same everywhere in the world, that the biomass of fish was unlimited, that if we didn't eat fish, we could walk on the sea. French philosopher, right? And this person, Marie Somerville, in the 1800s, was a graduate student from Oxford. She was not allowed to pass her PhD because she was a female in Oxford in the 1900 to be exact. And she was the first marine biogeographer of the world. What you need to remember of biogeography is basically the study of how the spatial gradient and the environmental gradient mix together in order to have biodiversity and to have species distribution. How can we understand why a species is there and not there? Why do we have more biodiversity here and less biodiversity here? That's biogeography. And she specializes in marine biogeography. And why is she so important? And it's a name that you need to remember. She has done that by going to all the ports in England. Remember, she's a graduate, female, scientist from Oxford in the 1900s. She's going to all the ports, Plymouth. I don't know, it's the English guys. And she noticed that depending on where the journey was, the catch composition was different. The species composition was different. So she started to map, okay, this species is from there, this species is from there. And after 20 years, she drew that map. And actually, that map still works. If the pre-Second World War was the discovery of the terrestrial new species, the post-Second World War was the time for the ocean. All the curve that you're seeing here, you see, it's increasing. We are discovering new species, then World War, we have other things to do, and then it's still climbing. This climb is drawn by all the discovery in the ocean, meaning that since almost 50, 60, 70 years now, we are discovering new species every year at a rate that is exponential. Unfortunately, it's an uneven focus. Why I'm saying that? We have some species that we like, and there are species we don't like. We tend to like our fish, the mammals, the whales, the dolphins. And if you look at the curve, this is the discovery rate of the mammalia. This little patch here will be the marine mammals. Look at the fish, how much they have been discovered in the last decades. But the problem that we have is that the invertebrate, we don't discover that much of the invertebrates. But the problem of that is that life in the ocean, from what we know, represents 1/3 of all biodiversity. So if you take the book of all the species that has been described since Darwin, 1/3 of them is marine. But the paradox of that is that this 1/3 represents 3/4 of the living biomass on Earth. On the graph that you're seeing here, each block is 1 gigaton of carbon, okay? This is us, this is the human. Here, that's the crustacean in the ocean. And here, that's everything that we are eating and the wildlife on Earth, on land. All that is the ocean. This is why understanding the life form of ocean is basically understanding our planet. We're not called a pale green dot, we are called a pale blue dot. Unfortunately, and that makes a lot of sense, we have a lot of spatial bias. When we sample the land, terrestrial, we have less restriction. Basically, we are terrestrial creature. We can go wherever we want, as crazy as you can be, like running 170 kilometers or not. You can go to the top of a mountain, you can go on the beach, and you can observe new species if you have the knowledge on species. When it comes to the ocean, you're dealing with a lot of difficulties. We are not marine creatures. We cannot breathe underwater. When we are going at sea, it costs a lot of money. You can ask my dean about that. And you're not dealing with two-dimensional space. You're dealing with three-dimensional space, meaning that you can go at sea searching for new species, you can put your net and be not lucky and miss it by 10 meters. So when you look at that map here, this is all the sampling of the last 150 years in a logarithmic scale, meaning the more it's slow, it's 1, 2, 3, 4, 5, and here we are talking in billions of samples. And you can see that we clearly sample always the same region. Coastal, why? Because it's easier to go. It costs less. Northern Hemisphere, why? Because unfortunately, historically, all the universities that were studying marine taxonomy or marine biogeography or fisheries or oceanography are in the Northern Hemisphere. So we have a clear, big lack on the Southern Hemisphere, which is paradoxical because where there is more ocean is the Southern Hemisphere. And on top of that, we only sample the subsurface, the first 100 meters, and what is near the coast here. In order to fix that, we have complex mathematical models to try to disentangle the problem of sampling bias and trying to understand an objective way to map the distribution of a species. I'm going to make it very simple. This is the ecological niche theory of Hutchinson. We have all experienced it. I'm going to make it very simple. The idea is that we have all a window of environment that we like. Imagine yourself when you want to go on holiday, okay? There is a place where you're going to say, I'm not going there because it's too cold. I'm not going there because it's too warm. I'm not going there because there is too much sun or there is too much rain. This creates a window. This window for marine species is not a window to go on a holiday, but to survive. And this is called the ecological niche model. The idea is extremely simple. We are taking all the place where we have seen the species, and we are matching where, what temperature, what precipitation, what amount of grass, what altitude we found the species, and we try to find that window, to model that window mathematically. And since at the moment that we have that window, we project it and try to see what is the probability of chance based on the environment to find the species now and in the future. In order to do that, you need a massive database. And as Dean Kurtman, so weird to say Dean Kurtman now. As Ben was saying, in order to be ambitious, you need to rest on giants. And the first giants were the FishBase database. The FishBase consortium was created in 1989, and the idea, they were the actual ambitious people. They said, we're going to create a website where we're going to catalog every marine species with every biological traits, every fishing knowledge, every distribution knowledge for everything. And they did it. And if you look on Google, and if you put any name of fish, that would be your first or second choice on Google. And if you look on the top right, there is a map. And that's actually us. They started in 2002, based on the model that I just described, to map the distribution of life, based on what FishBase collected. I arrived 20 years after, when most of the job was done, and it was pretty easy to go. And when AI arrived, so fancy models, and we create what we call AquaX, which is an AI pipeline that take the data from FishBase and take the best of the environmental data and try to map the distribution of all the life form where we have enough information to map. And we provide those information, you will see, to experts, to big consortium, to international collaboration groups. We have done that for 33,000 species. So this is all life form that we have in the marine realm. If it's black, that means that we have succeeded to model it. If it's gray, we still need to model it, or we don't have enough data yet to model it. So, in a nutshell, who are we? What is AquaMap? Who are we? We collect, we map, we inform. I took one simple example. This is the sand sea trout. All the black points that you're seeing here are confirmed presence of that species. And in blue, this is what the expert is saying where the species is. Okay? If you look at the East Coast, there is plenty of dots here, and according to the expert, should not be there. Why is there a presence of the species while the expert is saying it's literally impossible? We ran our model of this environmental window with AI, and we compute the probability to find the species. And you see that actually the probability, the environment is pretty suitable for the species. And we can zoom because our model can go up to 5-kilometer resolution. And so what you're seeing here, the end of that story is that the experts have drawn that map in the 1980s. And the climate change didn't stroke at that time. And with climate change, the species started to go in the Atlantic side and try to perform a poleward shift. And we captured that with the model. The environment is getting more and more favorable. And what you're seeing here is the projection by the mid-century, so 2050, in the worst-case scenario. If it's red, doesn't look good for the species. If it's blue, it looks good for the species. And you can see that the environment is actually getting better and better on the Atlantic side and worse in the Caribbean side, in the Gulf of Mexico. And now, this is the title of that presentation. I promised you to talk about biodiversity. If we can do that for one species, we can do that for 33,000 species. So now we're going to discuss about biodiversity. When you think biodiversity, the first metric you're going to think is species richness. How many fish, how many corals, how many mammals is there? That's the first dimension you're going to think. This is the map, and you're one of the first to see that. This is the map of marine biodiversity for 33,000 species at 5-kilometer resolution. You can clearly see that we have more species in the coast. It's not due to sampling. Yeah, potentially, yes, because we don't know what is in the deep, but we think that there is less species in the deep than at the coast for many ecological reasons. There is more species at the coast than there is in the open ocean, and there is more species towards the tropics than there is at the pole. And you can see with our higher resolution the effect of islands, like here the Galapagos, or here Iceland, or the Svalbard in the north of Norway, or the effect of the continental shelf in the North Sea. And we have done that for all those species. So we can divide those maps for reptiles, so turtles and snakes, for echinoderms, for fish, for mammals, for all the mollusks, so bivalvia, for example, and for the shark. But what is starting to be important is thinking about the other dimension of biodiversity. Think about what you want to preserve, what characterize biodiversity, and that would be rarity. Imagine you want to protect something that you remember from your childhood, something that is rare, that is precious. You want something that is rare. But rarity, how do you measure rarity? In what dimension do you measure that rarity? The first basic thing that you will do is spatial. Is it everywhere, or is it in only one place? And we did that. We computed how big is the distribution of the species versus how small is the distribution of the species. And this is the map of rarity in the world. The more it's red, the more the pixel, the region, hosts species that are only there. The more it's blue, the more the species are what we call commensal or cosmopolitan. They are everywhere. Think of tuna, for example. Like tuna really like open ocean, they are everywhere. Think of a blue whale. But when you start to come to small islands like Hawaii, like Easter Island, like Galapagos, or French Polynesia, you're going to start to see rarity. Species that have evolved and stayed only in that place. And look at the zoom on the Galapagos. While in species richness, there was a lot, but not that much. They were not a peak of biodiversity. In rarity, they are among the biggest in the world. And then you saw that there was a lot of species in the North Sea, but when it comes to rarity, not that much. There is a lot of species that have a huge distribution there. Then the last dimension you want to think is, okay, we thought about distribution, but what about the weird species? We all seen Nemo, the movie from Pixar. We all remember when the father of Nemo went into the deep and met the lanternfish. Like, the weird one with the little lantern? That's a taxonomically rare species. That means in the tree of life, he has his own branch. So he's not rare technically because he's almost everywhere in the world, but he's rare in his evolution pathway. Okay? So we map that. This is the map of the phylogenetic distinctiveness of the 33,000 species. The more it's red, the more they are weird creatures. I don't like to say weird. In a diplomatic way, they are evolutionary distinct. In a normal way, they're weird. Let's be real, they are weird. But in a more, and I don't like to think like this, but sometimes you need to think like this, in a more economical way. When you're thinking of potential patents, drugs, things that can solve cancer in the future, DNA that are only in that particular pathway of evolution, they are there. The more it's blue, if you notice, the coasts are blue, less rare. What does it mean? It means that the group that we have at the coast, they are very close taxonomically. They are from the same genus, the same family. And then the moment you're starting to go to the open ocean, the unknown, they evolve in a different way. They have different habitat. They are our Star Trek movie. So my postdoc, Yulia Igorova, that is here, has done a genius move one day. On a Sunday, she sent me that map. She said, I took the top 5% of all the metric. Don't forget about the big one. This is the top 5% pixels of biodiversity, species richness. So the highest in number of species. This is the top 5% most rare pixel in the ocean. And this is the top 5% of phylogenetic distinctiveness. And we mapped everything together. One analysis for what is in our coast, that's called the EEZ, exclusive economical zone, and what is outside the EEZ, called the ABNJ, area beyond national jurisdiction, called that the ICs. And you can see the hotspot of the world with all the biodiversity dimension. And what is fun is that when you speak about a hotspot in the ocean, the first thing that people will think is habitats. They will think, hotspot, coral reef, hotspot, seagrass, hotspot, the kelp, the seamount. Actually, that's not true. Habitats are a composite of the hotspot. And if you look at that graph, coral reef is holding most of the hotspot of species richness. That's true. But if you start to look at seagrass or kelp forest, they are holding the rarity. That means that if you protect kelp forest, or if you protect seagrass, you protect a different dimension of biodiversity. If you protect coral reef, you protect species richness. And when you go offshore, let's take the seamount, in proportion, they are protecting the phylogenetic distinctiveness. Unfortunately, and you all know about that, the ocean is under exploitation, plenty of exploitation. And we have a metric called the cumulative impact index, where we have pollution, fisheries, dredging. We have, if I do remember, 28 variables that we normalize and say, the more it's red, so in that case, the more it's purple, the more it's impacted. The more it's yellow, the less it's impacted. So very intuitively, you will understand that the coast is more impacted than the open ocean. And the pixels that you're seeing here are all the pixels where we think that there is a hotspot. And we divide that by continent. And what you can see is that most of the continent, if you divide the hotspot, are already impacted. The one that is the less impacted is Oceania. And it's the one that is the most protected. And at the moment that we are starting to go to Europe, America, or Asia, look at Asia. It is by far the most impacted by all human activities, and it is by far the less protected. And I'm sorry to say, but America, you're not doing great. And that gives us an idea. So the first model, I didn't talk about it, but the first original model from 2000 were at 1 degree resolution in a normal human-speaking way. 1 degree resolution means 100 kilometer by 100 kilometer. The new model that we have published are at 0.05, meaning 5 kilometer resolution. Okay? So this is the Galapagos. That was the resolution that they had 20 years ago. This is what we have now. Now we can inform management. Now we can say, go on that side of the coast, you need to protect it, or that side of the coast. While before they had, this pixel, it's the same. So what did we do? We have done? We have computed the biodiversity, all the metric. Species richness, rarity, and phylogenetic distinctiveness for every nation of the world. And all the region of the ICs. So that's the orange region, and you see the little circle here, that's the EEZ. And we have computed how much of the distribution of every species, so imagine tuna, or imagine cod, or imagine anchovy. How much the distribution of the anchovy, for example, is covered by marine protected area, an area where you don't fish and you're not supposed to pollute. This is a bit complicated. I actually hate that graph, but it summarizes everything. In the x-axis, you have the percentage of the unit, EEZ or ICs. How much is it protected? What is the percentage of that unit that is currently protected? And on the y-axis is the number of species in average that you will find, okay? Unfortunately, what you will expect is that the less you have species richness, the less you have protection. The more you have species richness, the more you have protection. This is not the case. This is actually not at all the case. So when Ben was talking about half Earth. If we have 10% of Earth, I'm happy with it. Like for now, look at the amount of nations that are between 0 and 1%. Here there is 158 nations. By the way, because I know everybody likes to check where we are, this is US. This is France, this is Canada. I had to do it. To simplify, let's simplify that graph because, again, I don't like it. If you divide species richness in 4 quartiles, okay? The lower species richness, medium, medium low, medium high, high. And you compute how much are you protected. You will expect the more there is species richness, the more you have protection. This is literally the inverse. It is in the place where there is species richness the lowest that we have the highest protection, and it is in the place where we have the highest species richness that we have the lowest protection. Same for rarity. The more the species is rare, you want this. The more you want protection. You want to protect where the species is, because it doesn't have thousands of places to be. And weirdly, we had one metric that was working, this one, phylogenetic distinctiveness. And it took me, I think, a month to understand why. It didn't make sense. Why all the metrics are failing, but not phylogenetic distinctiveness. And it's because of policy. And it's because of all of us. When we create protected area, we always try to focus on one group that we like. We're going to protect that place for that whale. We're going to protect that place for one seal. We're going to protect that place for one shark. And usually very distinct group evolutionary. That's why we are succeeding in the phylogenetic distinctiveness. And so that's called the umbrella strategy. I'm using a top predator that everybody likes. Easy one, you're going to understand in 5 seconds. Panda with WWF. The panda has been used to protect the forest. Unfortunately, it works on the terrestrial. It totally failed in the ocean. And why it's important to show you that graph is that our top predator, the one that we had, the emblematic species, are on the top of the food chain. Sharks, tuna, billfish, whales, seals. But if biodiversity erodes, if biodiversity extinct, the castle of cards is collapsing. And your top predator is going with it. And so, because I'm always the guy who's giving the good news, and I'm pretty sure you're going to be in depression after that, this is, for all species, how many species? So let's divide this. This is all the species in the world, 0 to 100%. Each color represents how much the species is protected. According to UN, United Nations, you need to have 30% of your distribution to be protected. That meaning that you're good, okay? Currently, 8.8% of the world is at that level. 8.8% of the species of the 33,000 make that threshold. More than half are not protected or barely protected. And when you go to the range rare, meaning the one that have the smallest distribution, it gets worse. It gets very worse. You're in the 60%. And the phylogenetic distinctiveness, look at that. 7% that are making the cutoff. That means that, yes, we have marine protected area. But are they doing the job? And of course, I always like to finish on a nice note. So let's go for climate change. You remember with my models, I can simulate the distribution, and I didn't went in detail, but I have the uncertainty, and I know where or when a model is failing or is too uncertain to be published. But based on this environmental window that I spoke about before, we can also simulate where it's going in the future, because we know what's going to be the condition in different scenarios of the future. This is the change of species richness by the end of the century for the worst case scenario. Red means doesn't look good, you're losing species. Blue means you're gaining species. But be careful, it's a mixed message here. What you're seeing is an erosion of the tropics. Why? The tropics are getting warmer. And the problem is not getting warmer. It's not the first time that the Earth is getting warm. It's how fast it's getting warm. Species don't have the time to adapt. They don't have the time to change their niche, their window. So they need to move. You have two choices when you're moving. Either you go deeper, the water is cooler. Either you change and you're starting to go to the pole. The problem with deeper is at some point, the deeper and deeper you go, the less and less oxygen you're going to have. The less and less light you're going to have, the less and less food you're going to have. So usually what you're seeing in migration, and not model, I'm talking about actual, real data, species tend to go deeper, they reach the place where there is no more light and so no more food. Once they reach that, they start to go poleward shift. And we saw that in North Sea, in Pacific Northwest, and in Nova Scotia. So we have an erosion of the tropics, and we have the Arctic acting like a refuge. But I told you, it's a mixed message. And that's very important to not simplify it. On the x-axis is how warm it's going to get. I'm sorry, I'm a scientist, and on top of that, I'm European. I'm talking in Celsius. This is how warm it's going to get. This is the Arctic region, temperate region, and tropical region. The more it's getting warm, as I told you, the more the species is going to go up in temperate. And the less you're going to have species in the tropic. On top of that, the habitat that they cannot shift. It's hard for a coral to have a dispersion. They have dispersion, but it's hard for them at the scale of climate change. Are disappearing. So the animals that are associated to this habitat are disappearing with them. Okay, let's go to the temperate. Yes, you're gonna be a winner. Yes, it's a refuge. That's very cool. I'm very happy with that. But that's not gonna be the same species that you're gonna have. You're gonna have what we call a tropicalization of your species. And put yourself 5 seconds as a fisherman in Nova Scotia, a fisherman in Seattle, that has been trained for a specific type of fisheries with a price on the fish that you're used and your ancestor have fished. And then all of a sudden, after 20 years, half of your catch is a species that was not there before. And more importantly, has no price. And the price, the value, is not worthless because you have your market, you have your consumer. What do you do? How does the fishermen are surviving that? Do we subsidize it? And then on top of that, you have the pole. And the pole, for the last 4th assessment of climate change in the IPCC, has acted as the refuge. We all said, okay, we are losing the ice, but at least the species is going to go there. Recently, and with my Ph.D. student, for X and Y reason, because of light, because of dispersion and currents, we are saying that it will not be a refuge. Why? Because you cannot put a tropical species or a temperate species that is, by evolution, used to seasons in a biome where you have 6 months of light and 6 months of dark. It's just not feasible. At least not in 50 years. Let's continue in the good news. This is the same but for rarity. What you're seeing here is that on top of the fact that there is less and less species, their range is contracting, meaning that all their distribution is contracting to where they can be. And only few refuges are left, very localized, and US and Alaska and British Columbia is one of them. And on top of that, you have an erosion of the phylogenetic distinctiveness in the place where we have the highest phylogenetic distinctiveness. I know, you've heard that kind of news like 1,000 times. I want to go back now in conservation. And we can act, we can do stuff. I just want to tell you where we are at now. So this is literally the same graph I showed you, with all species, range rare, and a high. If we keep the same network of marine protected area that we have now, and we let the species change their distribution with climate change, you agree that we have a fixed solution to a mobile problem. The number of species that are not protected or barely protected are increasing from 54 to 58%. And for the species that are range restricted, from 60 to 66%. So the one that we need to protect, if we don't reevaluate our marine protected area network, will be even more at risk. And the one that we thought were protected, again, we have a fixed solution for a mobile problem. So the ones that were in the marine protected area are moving, and the MPA is not following. And so we have what we call an erosion of the protection. And so what you're seeing is that by 2050, 20% to 50% of those species will not be in the category of the 30% protected. If we don't create corridors of climate change, we know where they are going. It's not quantum physics. A coastal species will use the coast. It will use the dispersion to go somewhere. If you create corridor of marine protected area to help them transition from one place to another, you protect the species. And we are not set up for that, for the simple reason that we are thinking nation by nation and not as a whole. So, to give you a final concrete example, all the maps that I present to you, that sounds very sciencey, very, and what if, what case. So, those maps are available on the website for everyone, to be honest. You go on aquamap.org, you will have those maps, and if you're a diver or a fisherman and you need the distribution of your fish, please don't destroy that fish. We can provide you those maps. For expert group, so for people who want to evaluate the stock of seahorse, the stock of tuna, they ask us the updated distribution of the species. You remember the example that I gave you with the sand sea trout, where the expert was in the 1990 and then you had to update it with climate change? We provide that currently, day to day. And finally, and I'm very proud of that, we published the data, the big map that I show you, and it was last November in the United Nations to set up the new marine protected area in the ice seas in front of Africa to protect the phylogenetic distinctiveness, the rare evolution. So, yes, it sounds super sciencey. Yes, we have nice numerical map. Yes, there is an application to that. So, what are your take-home message? This is very marketing, but I need to say it, like Ben. We deliver a 5-kilometer resolution. We have 32,000 species. It's pretty cool. My marine hotspots are multidimensional. Stop thinking number of species. One species that is rare can hold that as a hotspot. Our current protection is too low, misplaced, and does not protect the hotspot. Climate change is reshuffling biodiversity and eroding I have a problem with H and R. Irreplaceability, meaning phylogenetic distinctiveness and rarity. But we have solution. We have blueprint. We are testing different scenarios of corridors, of high seas protection. As you can imagine, this work I cannot do that alone. There is too many species, too much knowledge that you need. And so we operate as a consortium. So we are AquaMap, so the mapping part of a bigger consortium called FishBase, the FishBase consortium. And I cannot put all the face because we are 20 universities. So this is only for AquaMap, and we have University of Miami with these beautiful people here, University of Santa Barbara, University of British Columbia, University of Queensland, Dalhousie University, University of Hamburg, and Aquatics in the Philippines. And these are the core. I'm not even talking about the students and the interns. Finally, and this is very important and really time sensitive, all the work that you're seeing here since 1989, so from FishBase to now, nothing has been paid by federal grant. Not once. So I would like to thank our current funder, which is Conservation International and Environmental Defense Fund. It's because of these people that we do that work since 40 years. And it's because of donation. And it's very important for you to have, because usually people thank their funders and that's it. No, no. Without them, all the databases that are free and accessible to everyone in the world. If you're in Mozambique, Tanzania, Kenya, Vietnam, Sri Lanka, you have access to all the things that we are doing. It's because of those people, those foundations, when you give money to those foundations, that's the result that you have. And that's my phone. I'm not done. Most likely my wife. What do I want to say? Yeah. So if you want to donate, please do. You have Ben, you have all the team, Diane. Forget. Contact us and help us, because currently there is no federal funding, and let's be honest, there will be no federal funding for the coming next years. Finally, if you want to follow us on Instagram, this is the scan. You can follow us on Instagram, and you can email me privately. Otherwise, you contact Dean Kurtman. And I wanted to finish with your country. This is your species richness, and this is what's going to happen by the end of the century when you zoom on only the US. In that time, thank you.
Thank you, Gabriel. That was excellent. We have time for a few questions. Please wait for the microphone to be passed to you to ask your question. Hands, please, right here in the front.
Oh, thank you. Sorry.
You're first.
I am from the Lake Michigan area, and we do a little bit of mapping up there, but I'm wondering if your company has any connections with inland lakes or freshwater lakes around the world.
So I didn't talk about it because I had 1 hour.
Yeah.
We have done AquaFresh. We are rebranding it because the name is terrible. It sounds like a toothpaste.
Yeah. It's on your website though. Like
So we have done the distribution of 2,000 species of fish freshwater.
Okay.
Now and in the future.
Okay.
And it's in revision in global change biology.
Okay. And then
And if you want the data, we can collaborate. So you have lake and river.
okay, that's fabulous. Thank you. And then my one other question is, I do marine engineering, and I'm wondering if there is a type of installation, plankton, coral, or a species here for Florida that can increase the cooler waters here in Florida that you guys know about.
What do you mean? Do you mean like a set of combination of species that will cool the water?
So in Michigan, we installed biomimetic tiles on some of our inland lakes to increase plankton, and it decreased the water temperatures, which gave our trout more access to better food. And I'm wondering if there's a species here for Miami that we can do installations for.
So I'm a macroecologist, so I'm not a specialist of Miami.
Okay.
What I can say, though, 2 things. First thing, yes, you can always have a combination of species to buffer climate change. For example, we know that kelp and some species of coral species in the Red Sea can support high temperature and low pH. And that creates a shelter to all the coral species. That being said, I'm always scared to take one species in one ecosystem and bring it to a new ecosystem. You create a new dynamic in the ecosystem that is basically, I don't like to say that, but play God. And didn't went well often.
Hi, how are you? My name is Jose. I am originally from Ecuador, and I used to be the the Undersecretary of Fisheries there. And Ecuador is very important, a tuna fishing nation, right?
And anchoveta.
Mostly down south in
Down?
Peru, we are not that great there, but I'm thinking as a policymaker right now. And I can imagine the challenge that is going to be for the decision makers and the politicians to admit that they have to change the way they see their resources and how they are distributed in their waters or where they think that they can control. It's going to be a huge challenge.
Yes. So, how to answer that in a nice... We inform, we collect. No, we collect, we map, we inform. At some point, you need to be adaptable and to speak with people like you, trying to put something that sounds very complex with complex map into a simple number. And that's one of the jobs that we are doing when we are working with the United Nations. For example, working Ecuador and Costa Rica, or Ecuador with Chile or Bolivia, like to create a triangle of protection with the Galapagos.
Yes.
This is a perfect success of policy making.
It took 20 years to get there.
Yeah, but you
and
did it.
It required a hell of a lot of pressure on the private industry to allow that to happen.
Yeah, but you did it. Yeah. And you're what I call a blueprint of protection.
Good evening. Thank you for the presentation. I really enjoyed it. I learned so much. I'm assuming that the United Nations is the final governing body that you present this information to. Well, who do we report? Who do we seek information to or appeal to at the United Nations to say we'd like to learn more or we'd like to put pressure on this particular group so that these countries and the governance of these countries will make changes? Like what United Nations body is it that we...
I'm going to try to not enter in the political ring, because that's going to be a tough one. First, it's nice to have a country that is still in all the international agreements. For example, on biodiversity, it's called the IPBES, and for climate change, it's called the IPCC. Your representative in the Senate, in the Congress, in the Parliament, are voting for those, are agreeing and financing those. We report to them and to the world. So, depending on the question, for example, when I present my result, I can present my result at a state level, at a region level, at a country level, at a continent level, and then at a world level. This is the granularity that you have with the UN. To put pressure on the UN, you need to put the pressure on your state. And then after your state, on your government. To have international accord, agreement. A good example, the perfect example, the first example after Ecuador was the Paris Agreement in 2016. For the first time in history, all nations of the world said, there is climate change. We're going to do something. It's not, we all knew it was not feasible. What was important is that we agree to do something. By retracting, you destroy that idea. So when the current, I don't want to say it because I'm an immigrant. True. I'm sorry, but true. When US retracted from the Paris Agreement, so IPCC, US is currently the second biggest emitter of CO2 in the planet. Historically the first, and by a large margin. China is playing at your toes. They are coming back, they are playing at your toes now. You're retracting, you can do every effort in the world, in Europe and Africa. That will not even start to be one thing that America will do. Because you're the biggest. You're the biggest in emission. So your decision at a state level influences the world.
We have time for one last question here.
Thank you. You're probably not a policy expert, but I'm interested on your opinion on the on the High Seas Treaty
Yeah.
for biodiversity.
Did you-
And maybe follow-up question, do you see policies, country-wide policies working better or international policies?
Can you repeat?
Like country-wide, like within EEZs, do those policies work better when they work? Or is it better to have an international agreement?
I could do a whole presentation just on that. Okay, the ICs is a night. So that for everyone to understand, ICs as one rule is that there is no rule. Let's put it that way. It's the piracy rule, okay? What is in your country belongs to you, okay? So when we talk EEZ, the fish that are in your EEZ, they're yours. You can sell them, that's called quota selling, they're yours. When you're outside your EEZ, it's first found, first get. It's called the piracy rule, okay? UN is starting to create what we call the Horizon 2030 AB, lots of acronyms. We're gonna try to protect the ICs. True? We're good? Okay. The idea is the tragedy of the common. All the ICs belong to everyone, every individual on planet Earth, because it doesn't belong to a country. But since it doesn't belong to one person or one entity, it doesn't belong to anyone. It's the first one who first gets. So the treaty that we are trying to do is very complex, because if you want to protect something that doesn't belong to anyone or belong to everyone, who's going to put the money on it? We're good with that? So there is, have you heard about the Rashid Somaila idea, the BBZ? Okay. So Rashid Somaila is, I would say, the biggest economist on, what you would say, JC? Yeah, one of the biggest economists in the ocean. And he proposed something super childish, but works well. You take your EEZ, and you increase by 200 nautical miles or 100 nautical miles your EEZ in the ICs. Except this part become an MPA in the ICs. And because it's a prolongation of your EEZ, you're in charge of it. That will protect 27% of the world. So yes, we have pathways, ideas. Then, coming back to the politics, took them 20 years to agree between EEZ. What would be your guess if you wanted to negotiate in the ICs in the middle of the triangle? That is unfeasible. Because, just for example, USA just passed a bill, just 6 months ago, for deep minerals. First found it, first get. Nobody can protect it. No one. There is no treaty that can protect the deep minerals of the ocean. Remember, the deep ocean, that's where we have the highest phylogenetic distinctiveness. You mine there, you destroy that. And it will not come back. We're good?
Yeah, very good. On that very optimistic note. So I'm standing between you and the reception and access to Gabriel. So you might have lots of questions for him. I encourage you to talk to him personally. We've had 4 lectures this year, over 1,700 attendees, both in person and online. That's pretty impressive. Deanna wrote this for me, so I'm not going to take credit for it. So as they say in showbiz, that's a wrap. We're out. Please, enjoy.